Okay, guys, I'm going to record the rest of that lecture, at least try to anyway, here between classes on the cells that we didn't get through this morning. So I'm going to turn my video off and pull up the lecture on my screen. Okay, so we're ready to talk about osmosis, which is the diffusion of water. Diffusion of water through a selectively permeable membrane, which is what it states. In other words, it's diffusion of water through our plasma membrane. Water can pass through biological membranes, our plasma membrane, because of what we call aquaporins. Aquaporins are just a fancy way to describe our water channels. In order for osmosis to occur, there must be a concentration difference across the membrane, meaning we have to have high quantities of something compared to low quantities on the other side. And just like we talked about the other, or not the other day, but just earlier today, with diffusion of things like oxygen. Now it's a little bit trickier with water because in our body we obviously have water, but water's not alone in our body. It's a solution, meaning a collection of water plus other stuff, what we call solutes, dissolved in the water. Okay, so reading back through, osmosis to occur, there must be a concentration difference across the membrane, which results in the solution of one side of the membrane having a higher concentration of solutes or solute, and thus a lower concentration of water than the other side. Osmosis also needs a membrane that is more permeable to water than to the solute. Molecules of water will diffuse through the membrane from the area of high water concentration to the area of low water concentration. In other words, down its concentration gradient. So I kind of use this illustration a little bit more than the one that is on that slide, just to kind of explain this in greater detail. So we got an example here at the bottom, solution 1 and solution 2 with either 5% albumin for solution 1 and 10% albumin, which is a protein that dissolves in water. Now just looking at the figures here, you would probably just guess that this jar here is solution 1, this is solution 2. As you can see down here at the bottom, time demonstrates the fact that this jar is the same as this jar, just what happens after time. So solution 1 is actually this side of the jar. Solution 2 is this side of the jar, separated by a membrane, as it states, is permeable to water, but not albumin, which means that water can pass through, but the albumin is stuck on its same side. Okay. Now looking at the two solutions, solution 1 with 5% albumin, 10% albumin, have the same volume of stuff. Okay. I won't say the same volume of water because that's not actually the case. Okay. If instead of saying 5% albumin, this solution 1 said 95% water, and solution 2 said 90% water, that might help it easier to understand that this solution 1 actually has more water because it's got less other stuff in it, the other stuff being albumin, compared to solution 2, which has a greater quantity of albumin, which means it's got less water. Okay. Now, when you leave it alone and let time go by, water will travel from the high water concentration to the low water concentration, resulting in one, solution 2, that is, gaining volume, while the other loses volume, which would be solution 1. Ultimately, what takes place, though, and what's important, is we achieve equilibrium of the solutions as far as percentages go. Now, we have 7.5% albumin on the left, 7.5% on the right. Now, what this illustration does not show is the fact that this is an open-aired jar, which is not like a cell, because cells are closed containers. Okay, closed compartment or cell, a change in volume is accompanied by a change in pressure. Meaning, for example, if the water travels across and into the cell like a water balloon, if you continue to fill up a water balloon, pressure inside that water balloon is going to rise and rise and rise. Okay? This water pressure that develops as a result of osmosis into a solution is called osmotic pressure. Okay? Now, why I talk about this? Because it'll relate to what we call tonicity here in a second. Before I move on, instead of saying that water diffuses across the membrane from high water concentration to low water concentration, you can also state that water travels from low solute to high solute. So you can say it kind of two different ways: high water concentration to low water concentration, or low solute to high solute. So it's kind of tricky if you word it that way, because it's kind of the reverse of what we typically say, from low to high. All right, let's talk tonicity, something I just mentioned. As water moves by osmosis into or out of the cells, their volume either increases or decreases. A solution's tonicity is a measure of the solution's ability to change the volume of cells while altering their water content. So down at the bottom, we have three illustrations, and on the far right are three pictures of red blood cells, or erythrocytes, which is a fancy term, showing what happens when you expose those red blood cells, which are the same cells that you would see here in our cartoonistic pictures, when exposed to these different solutions. Let's talk isotonic solutions. Okay. An isotonic solution has the same potential osmotic pressure because they have the same concentration of impermeable molecules, which causes water to move in and out at the same rate. Okay, the solution that we're talking about is the fluid that's in the jar, and we're talking about comparing the stuff that's in that fluid, aside from the water, but what's dissolved in it. You know, if we just use salt, for example, what's in this solution compared to what's inside the cell, because that's a fluid with stuff in it, okay, salt being one of them. So basically, an isotonic solution is the exact same type of fluid with water and whatever dissolved in it as what's inside the cell. So there is no concentration difference, no concentration gradient. It means water is going to move across and in at the same rate, so no change in volume occurs. Okay. Whereas a hypertonic, the prefix hyper means high. It's like what happens if you put your cells in like salt water, and this explains why if you get trapped at sea, you're in a lifeboat, do not ever drink the salt water, because it's going to dehydrate you. Okay, because there's a lot of dissolved solutes in that fluid, and so the fluid that's in your cells is going to go to where there is a lot of solutes, like we just showed, from low to high, causing the cell to actually shrink. And such shrinkage of cells is called crenation, and actually looks just like this. This is a red blood cell shown with crenation taking place. What happens when you drink salt water is all that salt gets into your bloodstream, and then to try to dilute that salt out, we basically transport all of our water to our blood, which causes dehydration of every other cell, and that can ultimately kill you. So drinking salt water is going to defeat the purpose. Okay, hypo, that prefix actually means low. This is where essentially the fluid is like pure water, nothing dissolved in it, or maybe not enough dissolved in it compared to what's inside the cell. So there's a lower level of solutes than a living cell, causing water to rush in, so much so that it could actually swell and burst, like if you fill a water balloon with way too much water. And the term lysis means for a cell to burst. If you attach the prefix hemo, it refers to red blood cell bursting, okay, but it doesn't necessarily have to be a red blood cell for this to take place. Okay, so that's our forms of passive transport. Okay, once again, remember, passive transport is where we're going down the concentration gradient or electrical gradient or electrochemical gradient, and it's not requiring energy. Okay. Now we're going to talk about active transport. So some polar or charged solutes that must enter or leave body cells cannot cross the plasma membrane via passive transport because they would need to move uphill, which means they're going against their concentration gradients. In other words, we're going from low to high. When a transport protein moves a molecule against their free energy gradient, process is called active transport because it is an energy requiring process. And we're going to talk here first about primary active transport. And so during primary active transport, we utilize the energy obtained from the hydrolysis of ATP to pump something against or across its concentration gradient, okay? In other words, from low to high, okay? Anytime you see the word pump, It's going to involve primary active transport, okay, which means that we're talking about using ATP and we're going against the gradient. Okay. An example of that is the sodium-potassium pump, or sodium-potassium ATPase, same thing, okay, which pumps 3 sodium ions out of a cell, 2 potassium ions into a cell, resulting in a low sodium concentration and a high concentration, potassium concentration inside a cell. Okay. So looking at our figure, it's not 4 different proteins, but it's the same protein, and there's 4 steps to this process. Okay. Let's first note sodium. Notice we already talked about this in the lecture, but outside the cell, there's a lot of sodium compared to inside the cell. Okay. Well, with primary active transport, we're going to pump 3 sodiums from where there's low to where there's high. Okay. And we always use ATP in the second step. Remember that third phosphate is under a lot of energy, a lot of friction. We can transport that friction by way of that third phosphate to our pump, which allows for it to expel the sodiums in the outside, going from low to high. And then we can then bring in 2 potassiums. Now potassium is going from low to high into our cell. And then the whole process can start over again if we so choose to. Okay. Now the importance of this, we'll see a lot of this when we talk about neurons. But for right now, this process is going to drive secondary active transport. Okay. And secondary active transport, energy that's stored in ionic electrochemical gradients, are used to, or is used to drive the solutes or drive other solutes across the membrane against their concentration or electrochemical gradient. Because a sodium or hydrogen ion gradient is established by primary active transport, secondary active transport indirectly uses ATP or uses the energy obtained from the hydrolysis of ATP. So looking at our 4 illustrations of secondary active transport, we will not see ATP involved. Okay? The ATP involved for that process takes place and is utilized here in primary active transport. Okay? So what that means is, we know before primary active transport takes place, we had a lot of sodium out of the cell, very little in the cell. Okay, what we're doing is pushing what little we had out of the cell, which makes this area even more so loaded with sodium, and this area even lower with sodium, which means that we've created a even steeper gradient. And remember, the steeper the gradient, the bigger the drive to reach equilibrium, which is what we're going to utilize here. Okay. Most secondary active transport systems are driven by sodium electrochemical gradient. Secondary active transport, a carrier protein simultaneously combined to sodium and another substance, and then a change will occur to its shape, allowing both substances to cross the membrane at the same time. So we have a bunch, and even more than we did before primary, of sodium, and we're going to try to bring it in. And there are 2 ways in which we can do that through secondary active transport, either through what's called a symporter, which are what A and B are showing, or an antiporter, okay, which is what C and D are showing. In a symporter, when sodium travels down its concentration gradient, something else can take a piggyback ride, like glucose or an amino acid, and come in with it. But notice which way it's going. Okay. Glucose is going from low to high, which is demonstrating once again that we're going against their own electrical or concentration or electrochemical gradient. Okay. So that's what a symporter does, same physical direction. Whereas an antiporter is going to allow the substance to sneak out in the opposite direction, so leave the cell, same thing, from low calcium to high, or low hydrogen ion to high hydrogen ion, so against their electrochemical gradients. There is a third form of active transport. We won't really see ATP directly used in this process, but it is important to have a basic idea of vesicular transport, which is where we move stuff either in or out of a cell using spherical membranes called vesicles. And there are 3 forms: endo and exocytosis, which are the exact opposite one another. In endocytosis, what we do is essentially bring a substance into a cell. So think endo into, because the plasma membrane just kind of engulfs it and creates a bubble, a vesicle, bringing it in. Okay. So material moves into a cell in a vesicle formed by the plasma membrane. Three types exist: receptor mediated, which is highly selective, form of endocytosis by which cells take up specific molecules called ligands after binding to specific receptors on the plasma membrane. We don't really talk much about that. One that we talk about the most in this class is phagocytosis, which is what our white blood cells do to kill things like bacteria, even worn out cells, or even viruses. This is what we're showing here, and actually down here at the bottom, where our white blood cell engulfs, maybe it's a bacteria, maybe it's a virus, whatever. Same thing down here. There's actually a real microbe, zoomed in 450 times. White blood cell brings it in. And then, as we talked about earlier today, lysosomes inside of the white blood cell contain those enzymes. We can then fuse the phagosome that's formed when the substance is brought in, fuse that with the lysosome. The lysosome can then digest thanks to all those enzymes and break them down, leaving us with the digested material called residual body. Okay. There's also what's called pinocytosis, which actually means cellular drinking, which is different than cellular eating. Fancy term for pinocytosis is bulk phase endocytosis. It's called cellular drinking because it involves tiny droplets of extracellular fluid being taken up, and no receptor proteins are involved with that. We don't really talk much about that one. Now, the opposite of endocytosis is where that vesicle fuses with the plasma membrane and spits out the substance from the cell altogether. This illustration is showing you both endo and exocytosis. And so actually it's what it's showing you is transcytosis. Trans, which means across or through, is a combination of endo and exocytosis, where the vesicle undergoes endo on one side, and then moves the vesicle across the cell, where it then can go through exocytosis on the opposite side. This is what we do to move materials between our blood plasma and interstitial fluid, but we really don't talk too much about that either. Of all these things, really phagocytosis, which I always kind of think of like Pac-Man running around chewing stuff up, we talk about the most. All right, table 3.1 just is a nice summarization of all those different transport processes we just talked about. Last thing I want to talk about is the cell cycle, the process in which a typical or somatic cell duplicates its contents and divides in two. In other words, performing what we call cell division, the process by which cells reproduce themselves. Not all cells undergo cell division. For example, our neurons in our brain do not, but most cells in our body do, to replace worn out ones, dead ones, repair things, or to even just allow growth. Okay? Now, a somatic cell would be any normal type of cell is outside of reproductive sperm cell in men and reproductive egg cell in women. And those are called reproductive cells, not somatic cells. Okay. Two phases to the cell cycle exist, or two primary phases. Interphase, which is shown down here at the bottom, kind of on our pie chart, represents the majority of a life, or the majority of the life cycle of a cell when it's just normally functioning, whether it's a muscle cell contracting or a gland cell secreting, whatever, just doing what normal cells do. Okay. As far as cell division goes, there is a 3-step process in which we duplicate the DNA and get ready for the mitotic phase. Okay. The actual duplication or replication of DNA takes place within what's called the S phase of interphase, okay, which is where we take our double helix of DNA and we break the hydrogen bonds that exist between the two strands. So they say unzip it. So we have our two original strands. Now we attach a second strand to each of those, and we end up with two of the same thing. Okay, two copies of the DNA. Okay, So that's what takes place during S phase, so that we have twice as many chromosomes to begin the mitotic phase, where the nucleus divides, process we call mitosis. And now mitosis is specific to the nucleus, where the DNA is, but we also have the stuff outside the nucleus, and that divides in a process called cytokinesis, which kind of takes place towards the end of mitosis. You can remember the prefix, or I guess it's not a prefix, but PMAT, I guess I'll say. P for prophase, M for metaphase, A for anaphase, T for telophase. That'll help you kind of remember the correct order of mitosis. Prophase is where we first see what we call identical sister chromatids, the kind of X-shaped chromosomes, which we see here in our illustration over on the right side of the page. Back up here at the top of the page, we show a cell in interphase, looking in the nucleus. We talked about this. Most of the time, a cell is not in mitosis. It's not dividing, so we find the DNA unwound in the form of chromatin. So in early prophase, we finally see what takes place, where the DNA coils itself up and we see these chromosomes forming. We also see the formation of the spindle fibers or microtubules, things that look like little spider legs, traveling and trying to connect to the center, what we call the kinetochore of those chromosomes. During late anaphase, we see that the nuclear envelope has essentially dissolved. And now the chromosomes are just kind of free in the middle of the cell as they start to move towards that middle of the cell. Really, when the chromosomes are along the middle of the cell is metaphase. Okay? Metaphase, think middle for M for metaphase. And that's kind of the key thing that takes place there. We form what's called a metaphase plate. Here's what an actual cell looks like in metaphase with all those chromosomes aligned down the center. Anaphase is where those chromosomes pull apart, okay? And the cell begins to elongate. We even start to see possibly the formation of a cleavage furrow, which is the first sign of cytokinesis, where the cells are going to split into two halves, meaning that the cytoplasm is splitting. Okay, we definitely see cleavage furrow in telophase, when we also start to see the chromosomes dissolving back into the form of chromatin. We see a new nuclear envelope forming around each one of those. The spindles will start to disappear. And at this point, we essentially have two cells. We just have to break them free, which means that we have to finish the process called cytokinesis, division of the cytoplasm. Okay. And then after that, we end up with two new cells, should be identical to one another, and they are located in interphase at this point, ready for the whole process to take place again, which is amazing. Now, later on in the semester, we'll talk about meiosis or meiosis, which is a form of cell division that takes place in those cells that become our sperm and our egg. Actually involves prophase 1 all the way through telophase 1, and then a second round, prophase 2, and then telophase 2, where we end up with half the number of chromosomes. We'll wait and talk about that at the very end of the semester, because we'll learn about sperm and egg in better detail then. So it kind of makes sense to talk about it at that point. Last thing I want to talk about, this relates to cell division, is cancer, which is a group of diseases characterized by uncontrollable or abnormal cell division, which means that cells are dividing, and that causes the formation of what's called a tumor or a neoplasm, same thing, which is just a cluster of cells that can be very detrimental to our health. Okay. Cancerous tumors are described as malignant, which means that they have the capability to undergo metastasis, the spread of cancerous cells to other parts of the body. That's what makes them very dangerous, because then you can have tumors growing in many parts of the body and invading those tissues and getting in the way of normal organ function or tissue function, whatever. I've always been told benign, you're fine, because benign tumors are tumors that don't metastasize. Does that mean, benign tumors are safe because they're non-cancerous? No. Some benign tumors, like brain tumors that are benign, can actually be very dangerous as well. Now, going back to malignant tumors, the cells of those malignant tumors can duplicate rapidly and continuously. And what ends up happening are the cells then invade surrounding tissues where they trigger angiogenesis, the growth of new blood vessels. And that's because our body doesn't really understand and know that those cells are bad. They act like just normal cells, but what ends up happening is they compete with normal tissue for space and nutrients, which can often lead to death of the normal tissue. So what ends up happening when a person, eventually ultimately passes away from cancer, is that their body just shuts down because their organs don't function right. And that's because the tumors are just getting in the way and stealing all the nutrients from the good stuff. Okay, well, that will wrap us up on cells. So now you are good to go for the exam.